everyone. Thank you so much for joining me today. My name is Sudipa Chowdhury, here to present on the behavioral outcomes of early life caregiving with insights from the field of epigenetics. First, I wanted to introduce myself. I'm a rising junior at Newark Charter High School in Newark, Delaware. I'm a Delaware Hostess State Officer. I volunteer at a local children's hospital, and I intern at a behavioral epigenetics lab at the University of Delaware. I perform many procedures assisting in graduate research and replicating the earlier works of Dr. Tanya Roth, who I will be referencing today. Finally, I have aspirations of becoming an oncologist one day. So I wanted to first establish a common understanding of behavioral health as a field of study. Behavioral health involves the study of the emotions, behaviors, and biology that plays into one's mental well-being. Behavioral health is the preferred term to mental health as it encompasses the multiple contributions to mental wellness, including substance abuse, behavior habits, external stressors, and relationships, among other factors. Now, the question arises, should we classify behavioral health as a global health concern? When considering what constitutes a global health problem, we typically isolate diseases that pose a serious risk to masses of people from different regions, and illnesses that fall under behavioral health do in fact reflect the same pattern of high case rates paired with high mortality. These are the top behavioral health disorders recorded in 2019 with some of the highest global mortality rates in the millions. What is even more unfortunate is that these diseases can afflict anyone regardless of age or geographic location. From the same record provided by the World Health Organization, it was said that 1 in every 8 people globally have a serious mental health condition. So not only are these problems prevalent globally based on mere mortality or total case rates, but they also pose dire consequences, with many of these conditions leading to dysfunctional relationships, violence, poverty, substance abuse, and so on. I wanted to move our attention to early life caregiving so that we can eventually understand how it affects behavioral health. From birth to age 2, there is rapid cognitive development where synapses or connections between neurons are formed at 700 connections per second, a pace that exceeds the development rates of every other stage of life. Now, these connections are built throughout the child's experiences and through their environment. Therefore, the quality of a child's early experiences in terms of how sufficient their care is makes a substantial difference to their cognitive development, providing a foundation for learning, health, and behavior throughout life. Now, cases of child maltreatment during these critical developmental years yield behavioral health consequences. Children who face early life stress, or ELS, could have abusive caregivers, violent domestic environments. They may have food or housing insecurity, or have to cope with the loss of a family member, among other unfavorable situations. ELS is globally prevalent. For instance, international studies reveal that 3 in 4 children ages 2 to 4 regularly suffer physical or psychological violence from their caregivers. And ELS is generally known to hinder brain development, further impacting one's behaviors and increasing the risk of psychiatric disorders. Because neural connections are built in such unfavorable environments with insufficient caregiving, children will grow up expressing dysfunctional behavior and emotional instability. Now, knowing that a connection exists between ELS and behavior, it is important to further study this link, and this could be done through epigenetics, a study of the external or environmental factors affecting gene expression, which goes on to affect human behavior, health, and psychological development. One of the key mechanisms in epigenetics is DNA methylation, where a methyl group, CH3, as shown here, attaches to the cytosine nucleotide without actually modifying the DNA sequence, but instead turning off the gene it attaches to and preventing the protein from being synthesized or a trait from being expressed. We find that DNA methylation occurring in brain cells can actually rise from ELS, and this was the purpose of Dr. Roth's epigenetic study on the behavior of rats. We first identified the gene coding for BDNF protein, or brain-derived neurotrophic factor, which is a nutrient that enhances cognitive function, including learning, emotions, and behavior. The results revealed that methylation in the BDNF gene was much higher among the rats that were exposed to ELS compared to those who received normal treatment, as this first graph shows. So the gene was turned off and there was less BDNF available for the rat's brain to develop in a healthy manner. This second graph here shows the total amount of BDNF in both the prefrontal cortex and in the hippocampus. We mainly notice that there is a decrease in BDNF in the adult PFC of maltreated rats in comparison to that normal treatment baseline. Less BDNF further resulted in behavior changes, including anxious movement, hiding, or running away, which correlate to human behavioral conditions. Now, exactly how this research was carried out is simplified down to these methods and techniques. A rat model was used where, in a controlled environment, separate groups of infants were paired with mothers. Some received normal sufficient caregiving, while others were given few resources to offer care, so the mothers were more stressed and exhibited abusive caregiving. From infancy to adulthood, the behavior of the rats was observed and adult brain tissue samples were collected. Now, measuring methylation of the BDNF gene from the samples we received required two techniques. The first is bisulfite conversion, which I perform in this video here. The chemical bisulfite is added and is able to convert only unmethylated cytosines into uracil. That way, when the DNA sequence is later analyzed, only methylated cytosines will remain and can be quantified. Then real-time polymerase chain reaction, or RT-PCR, is performed to essentially isolate the BDNF gene we're targeting and amplify it so that we can readily measure methylation. So to summarize this topic, we classified behavioral health issues as global health concerns with many conditions arising from poor early life caregiving constituting early life stress. We can use epigenetic study methods, including DNA methylation observations, to see how epigenetic changes influence brain function and give rise to behavioral health illnesses. In terms of future applications, developing a more in-depth understanding of this essential connection between epigenetics and behavioral health will certainly increase the likelihood of treatment development for these serious behavioral health issues. Here are my references. And as I wrap up, I wanted to thank my mentors at the UD Department of Psychological and Brain Sciences for teaching and allowing me to work in their lab. Thank you to the Global Health Leadership Team for this opportunity to present on something I'm very passionate about. And thank you, the audience, for following along. If you have any other questions or want to come in contact, feel free to reach out. Thank you once again.